therapeutic drugs monitoring. The second drugs is called immunosuppressive drugs, which are used after the transplant has been done, because now these days the transplantation is increasing worldwide, and these transplant whenever your body receive any organs, the immune system naturally produce antibodies against that tissues or the organs, and that's why we need to provide a patient with the immunosuppressive drugs. And these immunosuppressive drugs are double-edged swords. Even the therapeutic drugs are also a double-edged sword, means their high level, other than the higher than the normal reference range, is also dangerous as compared to the low values, because if the values lower than the lower threshold, then the probability of organ transplant rejection is more. If the value exceed the normal range or a critical range, then it may lead to the carcinoma in some cases, the renal pathy, that's an renal dysfunction maybe leads, and sometime the liver complications are also maybe occurs due to this high dose of immunosuppressive drugs. So the third part is drugs of abuse there, that's called DOA. So in general, we can classify these drugs into three types. The first part is just give me. The first part is we call them a TDM, right? These are the therapeutic drugs monitoring. The other part is immunosuppressive drug. These are called IST. And the third part is we call them the TOA. That's the drugs of abuse, right? So on the basis of these three classification of drugs, we can classify all these drugs on the basis of their utilization. So let's start with the immunosuppressive drugs. So according to the feedback, the therapeutic immunosuppressive drugs, the most transplant, according to 2012 data, that's in the Australian region and in the North American region, especially in Canada, in America, and in these regions, they have a more transplant, and also in the European countries, due to the different organs failures, like heart, like kidney, like livers. So their transplant activity is more in these countries as compared to the developing countries. So these have a more potential to have this. That's why they have developed some immunosuppressive drugs, clinical trials, and they have few more clinical utilization of these therapeutic drugs. So let's talk about the immunosuppressive drugs. And these drugs are basically used in transplantation of human cells, tissues, and the organs. You can see that, and then they have to save many lives, basically to restore their essential functions, where they have alternate comparable effectiveness exist. So kindly, you have to make sure that this is and what the organ is usually transplant, and what type of immunosuppressive drugs are the choice drug for that transplant. So the kidney transplantation is by far the most frequent carried out transplantation globally. So even I can tell you that in my country, in Pakistan, especially in the city where I'm living, the Karachi, we have one charitable institute where the renal transplant, kidney transplant is done free of cost for the patient. And this is the one of the biggest institute in Pakistan, so providing a free of cost renal transplant. But the important thing is that the donor should be from the blood related or a family, because the the tissue matching and HLA typing is more compatible if you have a donor from your blood relation. So that's why they are creating, avoiding that market, black market for the organ donors. That's a selling option. That's why they are preferring if the donor is belongs to the same family. That's why they are using that. So this is the most important common organ transplant as of today. That's a kidney transplant. So if we talk about the transplant types, The global have a heart, livers, kidney, and the lungs transplant. If you talk about the survival rate of such patients who have these transplant, among these, you can see that from 1 year to the maximum 5 year survival rate. If we talk about the highest rate of survival of 1 year and the 5 years is in kidney transplant. You can see here it's a 97.2% in a 1 year, and in the 5 year it's 87.7%. So then after that, there is a survival rate of more in the liver. Transplant is in around 90.5% of 1 year, and the 5 years up to 77.8%. And then the heart transplant and the lungs transplant. So these are the four major transplants occurs globally, and the survival rate history from 1 to 3 years and 5 years. So let's talk about the immunosuppressive drugs. So, what is the function of immunosuppressive drugs? So, if we talk about in details about the immunosuppressive drugs, these are helps to prevent the rejection of the transplanted organs and the tissues. So, usually in the heart, lungs, liver, and kidney. These are the foreign organs, or even from the donors. Maybe they have a HLA typing is exactly matched, everything is perfect, but when you transplant any organ and tissue in the body, the natural immune systems trigger, and then they create the antibodies. Just to suppress that immune systems, just to prevent the rejection of this organs, transplanted organs, the physicians prescribe the immunosuppressive drugs. So these immunosuppressive drugs treat the autoimmune disease as well, like rheumatoid arthritis, myasthenia gravis, systemic lupus erythematosus, and they also treat some of other non-autoimmune inflammatory disease like long-term allergic asthma control as well. So these drugs are basically have an steroidal impact, or sometimes they have an immunosuppressive impact that make helps to prevent from the any immune system adverse effect. So if we talk about the immunosuppressive drugs, these drugs are usually called immunophilins. And these immunophilins are usually used in the market is FK546 and CSA, sirolimus, everolimus, mycophenolic acids. So these are the well-known immunosuppressive drugs as of today. The journal name for FK546 is a tacrolimus, and the CSA is cyclosporin. I can write here. Just give me a 1 second. This name is this is tacrolimus. Okay, and this drug is called cyclosporin. Right? So these two drugs are the most common drugs is the cyclosporin and then the tacrolimus, and then in some countries, mycophenolic acid, sirolimus, and everolimus. And the utilization of these drugs is depends upon the tissue and the drugs and the clinical history of such patient who has been used in the past, and the physiopathology of that drugs. So this is very much a drug of choice for majority cases. Cyclosporin, then tacrolimus, then sirolimus in mycophenolic acid, and then everolimus. So these are the sequence for the drug utilization. So let's talk about individually about the one by one. So the first drug we have to discuss about is 5K546 um, tacrolimus. So if we talk about the tacrolimus, it's basically a 23 members macrolide lactone, and that was first discovered in 1987 from the soil bacterium Streptomyces tissucobanensis. So this is the bacteria which from which we have discovered this macrolactone, and that was first approved by the Federation for Drugs and Regulatory Authority in 1994 in use in the organ transplantation. So this is the utilization of 506 FK. And if we talk about more about the tacrolimus, it's a basically a hydrophobic macrolide antibiotic, and this is basically in a high absorption rate and variable between the individual and maybe influenced by the structure. So you can see here in this structures, they have a very important information about that, about their structures and their about the absorption rate about that. So the HA has a half life of 4 to 41 hours. So what is a half life? If you are taking a 20 milligram drugs and it's become a 10 milligram in how much time, right? So if you are taking a, for example, suppose if you are taking a 20 milligram drug, or you have uh, in how much time it's become a 10 milligram in your bloodstream. That's called a half-life of that drug. So the major part is in a peak level and the trough level of that drug. This is also an important in this therapeutic drugs monitoring as well. So if we talk about the cyclosporin, that's called CSA. So this cyclosporin is a natural product, and that's produced in from the fungus, basically a Tolipoclidium inflatum, and comes in the medical use in 1983. It was isolated in 1971. According to the World Health Organization, listed for the essential medicine, this is the one of the drug of choice for the renal transplant and other organ transplant in the world. So if we talk about the variability of this, bioavailability is variable, and then the metabolism is also occurs in the liver as well. The excretory is the bilatory, and the half-life is about 24 hours for this cyclosporin A. So the important thing is that for the all immunoassay suppressive drugs, they are not found in the free form of the bloodstream. So they are tagged with the RBCs. So usually what the manufacturers of in the IBD market, what they have done, they have a manual procedure for the treatment of that RBCs with some special reagent, and then they after extraction they put that extracted or supernatant samples with the reagent and then react it with and then gives you the results. So they are tagged with the RBC. That's why in a mostly cases the sample type either the cyclosporin or a tacrolimus. The sample type is purple top tubes, and these purple top tubes are to acquire a whole blood. The sample type is the whole blood for the immunosuppressive drugs, and then they have to be treated manually or automatically on the instrument to give you the results from that product. So if we talk about more about the FK506 and the cyclosporin, both the tacrolimus and cyclosporin, how they works. So the major function of these type cyclosporin and that they impact has an T cells receptors, and they primarily to block in the transcription of interleukin 2 messenger RNA. And then the calcium channels are activated and the CAM and the calcineurons are activated, and then it's IL-2 gene activation, then nucleus. So this is the basically the action of cyclosporin and tacrose limits in immune systems of the lymphocytes, especially the T cell receptors, binding protein, that's the and bonding protein as well, CAM and the gammodium. So this activity has helps to stop the immune system to respond against that. So why get this test is this is very much important. Why we need to get test the tacrolimus and the cyclosporin levels, because this is a double-edged sword, and there is a very narrow therapeutic index range for these drugs. immunosuppressive drugs, they have a large inter and intra-subject pharmacokinetics variability, and their incorrect concentration in the patient blood could result in the transplant rejection if it's lower than the reference range, or if there's an accumulation of the toxic effect. So this is a double-edged sword. So if this is a range for, like for example, 5 to 20. So if it's higher than 20, it may lead to some toxic effect. If it's less than 5, it may lead to the transplant rejection. So this is very much important to keep this cycle balanced. Otherwise, individual high dose or a low dose may lead to some complication, either in terms of toxic effect, either in terms of organ transplant rejection issues. So these therapeutic drugs monitoring is usually employed or facilitate tacrolimus sacrolimus than the titration as well. So if we talk about more in case of side effect, if the value exceed than the normal value, so either the tacrolimus or the cyclosporin, they may lead to the nephrotoxicity, may have the toxic effect on the nephrons of the kidneys, and they have may impact problematic impact on the gastrointestinal tract. They may also disturb the GIT inner lines, and then may leads to some other complication which are GIT tract related. And they also may lead to, due to the toxicity, hyperdose of that immunosuppressive drugs may lead to the hepatotoxicity. And then if the drugs levels are not monitored properly, or if they decrease or increase the value, so they have the risk for type diabetes, risk of having taking insulin as well. So these are the side effects of having high dose impact on different organs like GIT, kidney, liver, and then the beta islet cells of pancreas, to not to produce the insulin, and then the risk of type 1 diabetes may occur. So if we talk about more, what are the other than the organs toxicity, they may also lead to some hypertension. In such cases, when the patients are on immunosuppressive drugs, they may develop the hypertension, and the carcinoma risk analysis is more. In such patient, they have a high side effect. If we talk about the psychiatric problems, then these side effects may lead to that. So if we talk about these complications or a side effect, when they stop the using of that drugs, or if they maintain the drugs levels, so they can be saved from these toxic effect on the organs, and as well as these side effects of hypertension, cancer risk, and neuropsychiatric problems as well. So the therapeutic drugs monitoring. So the therapeutic drugs window is basically in a range of doses that produce the different therapeutic response, having a significant adverse effect in the patient to provide the most effective dose with the least possible toxicity. You can see here, this is the time graph between the time and the plasma concentration when you give the dose of any drugs that go to the up to the peak levels, and this peak levels acquire, and after that is the sub goes to the subtherapeutic range, and this is the therapeutic window. So this is the therapeutic windows. If lower than this value, may have also adverse effect. If higher than this value, may affect adverse effect, and if it's lower than this desired response, then the subtherapeutic conditions occurs. So that may leads to also some other complications or some other cases in the therapeutic drugs as well as in the immunosuppressive drugs. So the minimum toxic concentration and the minimum effect concentration is equal to the minimum inhibitory concentration. So this is very much important. whenever you are monitoring either the therapeutic drugs or immunosuppressive drugs, the both monitoring the adverse response and the desired response. We need to keep the reference ranges for or the patient value ranges among the reference ranges, this therapeutic window, right? So this is very much important for the therapeutic drugs window to have some monitoring for that. If you talk about the large variability between the patient, yes, you cannot judge the dose and the organs with the patient dosage value. Different people have a different response in such cases. So everyone who has a transplant, they're monitoring for immunosuppressive drugs, or the patient if you're on a treatment, the therapeutic drugs monitoring of TDMs that need to be monitored with more frequently, and then on the basis of that dose, on the basis of that results, and the physicians prescribe or increase or decrease the dose of the therapeutic or immunosuppressive drugs. So the multiple influence factors that may matter the drugs level in the blood, and there is a no proportional ratio between the drug dose, drug level in the blood. So again, a person to person vary. So the drugs level monitoring is very much important in the patient. Like if we talk about the tacrolimus concentration of in a time profile from 0 to 12 hours duration in a different 78 lungs transplant patient. You can see in a some patient, the peak levels is acquired between 0 to 2 hours. In some patient, the peak level of the drugs is acquired at between 2 to 4, and in some cases, the peak level is acquired at between 5 to 7 hours after the administration of that drug. So this is very much important. There are multiple influence factors, the genetical factors, and the immune response has a different response against that immunosuppressive drugs, and the monitoring is more essential. So if you talk about the pharmacokinetics models for that effect, like absorption, if we talk about the absorption, you can see here the oral administration of or intravenous injection. They have high fat and carbohydrate meals may also have an impact on the absorption rate of that drugs. If you talk about the distribution, the binding of the immunosuppression to the blood proteins are pivotal variable, which often change early after the transplantation. They have also an excretion mostly in the feces and less than 3% in the urine, or operation in the digest tract, and the diarrhea may occur that may lead to the excretion of that. If we talk about the metabolism, that's maybe some inflammation, ischemia, perfusion injury, the cross reactivity with the metabolites and drug to drug interaction is also the factors which may impact on the pharmacokinetics model of immunosuppressive drugs. So, if you talk about the organ transplant patients which have administered the multi drugs, this is a real story. When you have a renal transplant, you are not taking only a one drug; you are taking multiple drugs for the different handling situation. So these drugs may situation of the human body drugs, and they have a cytochrome P450 enzyme to deactivate the foreign organic molecule like toxin and the drugs. So this process may lead to the drug-to-drug interaction. So this is also a big challenge for the chemiluminescence technique developed that their drugs-to-drug interference keeps them in a minimized ways as compared to the LCMS, which is more still a gold standard for the monitoring of therapeutic, immunosuppressive, and the other drugs. So let's talk about the therapeutic drugs monitoring. So ideal routine therapeutic drug services, if we talk about the ideal routine therapeutic drug services, the same day reporting is available, and they have economical, robust, and reliable access. How you can say that it's a more reliable that if you are running on a consistently on a reliable analyzer, this is more robust and the reliable in terms of quality and performance wise and economical in terms of the wastage wise. If we talk about the wide measuring range, ideal routine TDM services should be having a more big measuring range. If they have a big measuring range, that helps the patient or the physicians to have one results within a less time. The second thing is that the measuring range is, if it's the broad, it can help the patient for the monitoring in an accurate way rather than to have a dilution with the factors, and also they have a dilution factors for that. So the other thing about the capability to analyze the emergency samples rapidly. So this is called a prompt response from the diagnostic laboratory to provide a more reliable results, to monitor the toxicity or a bad impact or organ rejection in case of immunosuppressive drugs, and if in case of therapeutic drugs monitoring, to increase the drugs, therapeutic drugs, and decrease the value, decrease the dosage of that therapeutic drugs is more important in case of even in an emergency response as well. So if we talk about what are the methods considerations based, all the therapeutic drugs monitoring, immunosuppressive drugs, and the drugs of abuse, all they have some method consideration which is depends upon the sensitivity, which depends upon the accuracy, then reliability of the assay performance in terms of negative predictive value, in terms of positive predictive value. Along with that, the selectivity, the method that what they were using, that it's an tedious procedures or a fully automated procedures. If we talk about the sample requirement, this is also one of the most important type, when the sample type is either a serum plasma or a whole blood, or that whole blood samples are treated manually or an automation on the analyzers stood for that. If we talk about the clinical useful turnaround time, which is the most important part when you are monitoring either the therapeutic drugs, immunosuppressive drugs, or the drugs of use, because in a turnaround time is very much clinically important when the emergency patient samples are treated on that. And the cost is also a big factor as compared to the LCMS. The cost and the tedious procedure both are in terms of CLIA technique is more reliable and more easy to use as compared to the LCMS. So if we talk about the method of choice, the most prevalent approach in the clinical laboratory is the antibody-based assay, like immunoassays, to give the solution to the customers. So if you talk about the LCMS, basically chromatography-based assay, liquid chromatography, or a tandem mass spectroscopy is the in comparison with that. So if you go for a further in details, you can see here the mass spectroscopy is basically a tedious procedure. How it's a tedious procedure? First, you have to patient samples, then you have to prepare the samples. After preparing the sample, you have to inject, and then the separation technique on the basis of the liquid chromatography, and then this separation technique, the ionize and the mass analyze one, then the fragmentation is done, then mass analyze two, and then data analysis and the report generation occurs. So this whole process of the high performance liquid chromatography device take a tedious procedures, expertise, time consumption, and the cost is costly. So you can see that then a mobile phase, then sample multiple component mixtures are going there. Then they have some mass spectroscopy, some different troubleshooting for that, and then detections occurs, and then chromatograph, mass spectrum analysis occurs to the software. So this is a two major part. One is the analysis, that's in sample preparation, and then the detection. So these three major parts of the mass spectroscopy are very tedious procedures, and the expertise are required for that such systems. So if we compare the method of choice with the immunoassay versus the LCMS, the most important point for the immunoassay is the most widely used method in the routine laboratories right now these days, and they are easy to use. There is no specialized operation is required for that. Their integration in the laboratory system is easy along across the platform, from small instrument to the big analyzers. They have a shortest turnaround time, and in terms of labor, in terms of utility, and in terms of purchase cost, we are the most cost effective in terms of immunochemical technique base. We have a cross reaction with the metabolites It's a challenge in immunoassay. The probability of cross reactivity with the metabolites and then the other drugs is also a challenge. And then you have to be more selective to pick the right molecule when the immunoassay techniques are using. So we have interference for the endogenous compound like heterophilic antibodies, which is very common in the immunoassay analyzers. Or if we talk about the LCMS, they have a high sensitivity and specificity. They have a capable of wide linear range, and they have alternate matrices like dried blood spot, peripheral blood mononuclear cells, and tissue from the transplant organs to detect this drugs level in that. So it's required basically a specialized strain specific staff. That's a complication of the use. More expertise are required, and then the results from the different laboratories are not necessarily comparable. So they are highly sensitive and specific, but they have a challenge of imprecision in the LCMS because even when you have a lot of manual stats, when you run the same samples in 2 different days, you may get the variation in the results because it's all depends upon the manual handling of these samples on the LCMS assay. If we talk about the space, yes, for the LCMS, a separate space and then special requirement is for the LCMS. So this is also a big challenge for the lab now these days, where the new concept of throughput is meter is a test per hour per meter square is introduced. So the long turnaround time, it's also a big challenge in LC-WMS, and they have expensive analyzers and the reagents just to meet the customer's expectation or who want to run that. So in the immunoassay, there's a one more challenge. It has been reporting showing that the immunoassays are estimating, showing estimation, overestimation. If we talk about the cyclosporin, that has been estimated that between 11 to 57% higher estimation or overestimation of immunosuppressive drugs in some patient. In the tacrolimus, there are 17 to 18% are reported in some patient with have a overestimation of tacrolimus. These are the reference ranges. These are the references from where we have collected that data of 11 to 57% in cyclosporin and 17 to 18% in tacrolimus. So these are the lines where we can see that where this has been published in the journals as well. So if we talk about the cross-reactivity in immunosuppressive drugs and their metabolites, you can see here in the cyclosporin values, you have a major, their metabolites are AMI, AM1, AM9, and the minor are AM4 and AM19 and AMC9. So in this tacrolimus case, these are the metabolites which can interfere in the cross-reactivity impact on these assays when you are detecting these molecules. So the major limitation of immunoassay are significant cross-reactivity with the various metabolites of the patient parent's drugs. So this is very much important cross-reactivity, how we can minimize in that, we will see in the next slides. So the difficult for the antibody use in immunoassay to recognize only the parent drugs without any metabolites, the cross-reactivity because of their similar molecular weight and similar structure. So this challenge is due to the same molecular weight and the structure. They can react with the target reagent molecule, and then they gives you the falsely elevated result in some cases, and then they have to give you difficult to handle the antibiotic use in the immunosuppress to recognize only the parent drugs. So this is the challenge of a cross-reactivity in immunoassay technology. So in some cases, when they have compared the immunoassay, performance with the LCMS. You can see that the LCMS is graph A of tacrolimus is okay, but in some cases there is overestimation of the immunoassay due to the drugs interference. So you can see here there is a drug interference, and the cross reaction with the metabolites. So this is the most challenge in the different samples when you are overestimating in the immunoassay systems. So the mean predictive error for the different immunoassays methods, according to this mean difference immunoassay spikes concentration They have tested this spikes concentration on the chemiluminescence immunobase, ACMIA, EMIT, or ECLIA as well. So you can see here this is study involved 107 Japanese hospital laboratories, routinely involvement in the therapeutic drugs monitoring of immunosuppressive drugs of tacrolimus. And they also checked the spike sample did not include these metabolites, and then they check it. You can see that the ECLA has also a variation and the variability in this technique, and then they have other assays also have a problem with that. So here you can see that this value of CLIA is increased, and then you can see here. So with that, with this dotted line, you can see that the mean predictive error for the different immunoassays is existing. So that's the challenge. So for the requirement for the limit of quantitation, according to the different companies' guidelines, it has been reported that the proposed target through the level of tacrolimus in the whole blood is between range between 2 to 4 nanogram per ml, which is also an assay with the limit of quantitation below this range. So the SNIBE has also meet the expectation of the requirement for the tacrolimus whole blood, that because we have a limit of quantitation in 0.625, which is lower than 2.4 to 4 nanogram per ml. So if we compare the cyclosporin of Architect 2000 1000, they have upper 95 percentile is 0.5 to 0.8. If we talk about the dead bearing ACMIA technique has a lower value of 2.5 to 3.12, which is basically in the range of the tacrolimus whole blood proposed target values. So if we talk about the EMIT or Viva E, they have a 2.8 to 3.2. And if we talk about the Alexis, they have a 0.2 to 0.3 value for that. And if we talk about the QMS, they have a 0.7. So as of today, we have a very good internal performance validation report. On the basis of that, we have a limit of quantitation is 0.625, which shows an excellent lower detection value within the limited allowable error of these assays. So if we go further for challenges of the therapeutic drugs monitoring, the first challenge for the therapeutic drug monitoring is the clinical need consistent results and have a clear relationship between the drug level and the clinical outcomes. So this is most important, and it's always recommended whenever you are under the treatment for any drugs and you are on a therapeutic drugs monitoring, you have to prefer always a one lab which you are going for a baseline sample and the follow-up samples. So this is also important for each patient when they are testing that therapeutic drugs, either the immunosuppressive drugs or the therapeutic drugs for that. So is the existence of the multiple analytical methods with the variable performance? Yes. Multiple analytical methods have a variable performance. That's why it's always recommended for the transplant patient and for the patient who are on a therapeutic drugs. They have to keep one instrument as a reference instrument for their future monitoring of the therapeutic drugs or the immunosuppressive drugs. So if we talk about the challenge, another challenge, that's a lack of reference method and the reference calibrators are there. That's why the therapeutic drug monitoring is one challenge. If we talk about the LCMS, that's a gold standard for the monitoring many drugs, but there is no standard method also for that. LCMMS is existing. If we talk about the choice of method of immunoassay or LCMMS depends on each lab characteristic like workload, staff qualification, and the cost. These are all among the challenges of these therapeutic drugs monitoring. So let's start what SNIDE is providing solution from that platform. So any question? Okay, Don, thank you. So let's talk about. Okay, Rina, thank you. So if we talk about the meglumine therapeutic drugs monitoring assays, So what we have. So as you know that we have two generations for each analytes. If you talk about the FK546, which is a tacrolimus, right? So we have a first generation and the second generation, and you know that the second generation is a more improved as compared to the first generation in terms of traceability, in terms of sample type, in terms of calibration on board stability, in terms of sample volume, in terms of LOB, LOD, and LOQ, along with the linearity, and in terms of onboard stability as well, and the QC options which are we are providing from the free of cost from that platform. So if we compare the first generation of tacrolimus versus the second generation, which we call them FK546, we have both generation. We have a 50 test spec size and 100 test spec size, and we have a competitive and quantitative detection principles. We have a samples type of whole blood. If we could talk about the traceability, the first generation was traceable with the internal reference substance from the SNI, but now we have a cyclospore tacrolimus with the USP reference standard method, which is according to the global guideline for the standardization of K546, and the next generation is also in the second year. So if we talk about the calibration stability, now we have a 28 days stability as compared to the 1 week calibration stability. We have a same sample volume, but in the second generation, we have an improved sample aspiration volume from 40 microliter to the 10 microliter. In the limit of blank detection and quantitation, we have introduced the value of 0.5, which is a limit of quantitation, and the linearity in the previous generation kit was 0.1 to 100 nanogram per ml. Now we have a linearity of 0.5 to 50 nanogram per ml. So onboard stability is a 1 month in both cases, but in the second generation, we have an edge of two levels of QC. These are low and high level of QC. So the second option, if the customer wants more than two level of QC, they can ask for a third party quality controls from even from maybe from the SNI platform, either from the BioRed or the Randox platform. So if we go for a further comparison of 5K 546 first generation with the competition, so you can see here, if you compare with the Abbott, Roche, Siemens, and the SNIBE, they have a competitive principle, and SNIBE is also a competitive principle. For the reference ranges, for the Meglumi, FK546 tacrolimus assay, they have a reference ranges of Abbott, Roche, Siemens, and the SNIBE. There's no firm therapeutic ranges defined that according to the lab and according to the transplant institution. They have their own set ranges. If we talk about the limit of blank detection and the quantitation, you can see that we have an improved measuring range from 0.1 to 100. And if we talk about the limit of blank, limit of detection, we have a good limit of blank as compared to the other assays. So if you talk about the sample volume, we have just 40 microliter aspirating whole blood sample volume for that assay. If you talk about the sample type, the whole blood sample type is recommended because these drugs, which are FK5 or 6, are not free, found free in the bloodstream. They are tagged with the RBC. That's why we need to pre-treat the whole blood with some reagent to remove these 5K546 drugs in immunosuppressive drugs in the human blood. So if you talk about the calibration frequency, the change in the lot and after 38 days and 7 days and 10 days and 64 days are recommended in the Siemens and Roche. We have an onboard calibration stability for the first generation was 7 days. Stability on the board were 30 days, which are equally good as compared to the other, except the Roche, they have a 56-day claim for the onboard stability of that reagent. If we talk about the test pack size, this is a 200 test pack size the Abbott has been providing, while the 5K 546 is a 100 test pack size. They have a 400 test pack size. We are the only company who's providing a 50 and 100 test pack size for this 5K 546 first generation SM. So if you talk about the pack size, the pack size is 200 test. We have a Roche has a 100 test. The Siemens has the 400 test pack size, and the Snipe has the 1500 test. We have a free of cost calibrators and control included in the reagent, while the other competitors like Abbott, Roche, and Siemens, if they want to run that assay of tacrolimus, they need to buy separately the calibrators and control for such assays, and it's then a big challenge for that. So if we go for 5K 546, second generation comparison with the Abbott, Roche, and Siemens. So as of today, we are having this F506 that's in competitive principles. Among that, they have no firm therapeutic range for that. The measuring range in Abbott, Roche, and Siemens is compared to the Snipe. We are in a good position because we have for the upper side we have a good measuring range of 50 nanogram per ml. So if we talk about the limit of blank detection and quantitation, we have a very comprehensive LOB, LOD, and LOQ value. We have a minimal sample volume, and the sample type is the whole blood. The calibration frequency is 28 days, 1 month, and the stability on board is 1 month. So the pack size for us is the 50 test and 100 test, while they have a 200, 100, and 400 test pack size. And we are providing free of cost calibrators and control, while the other Siemens, Roche, and Abbott customers, if they want to run the tacrolimus on the instrument, they need to purchase calibration controls separately. So this is the comparison of first generation and second generation tacrolimus with the Snipe tacrolimus. And if you talk about the Meglumi FK506 first generation, according to this data, of method comparison between the Snipe Meglumi FK506 first generation in results in nanogram per ml with the commercially available 5K 506 assay. You can see that on the basis of these patient results, they have run 100 samples, and on the basis of these clinical samples, they have shown excellent correlation through the regression value of 0.99, and the patient value is less than 0.001. So this is an excellent correlation of our first generation assay. And if we talk about the second generation of assay, this is also correlated with the other competitors in the market as well. So if we talk about CSA, as you know that the CSA is stand for the cyclosporin. So the cyclosporin kit, we have a first generation and second generation, both have a 50 test and 100 test pack size. If we talk about the detection principle, they have a competitive and the quantitative analysis in terms of cyclosporin. So if we talk about the sample type, the whole blood type is recommended. If we go for which tube is recommended, so either a dipotassium or a tripotassium EDTA tube is recommended. Tripotassium EDTA tube is recommended, right? Or this tube may have a cap color is purple. So the cap color is purple is means this is either a dipotassium EDTA or a tripotassium EDTA, right? So it's up to you that according to the package insert recommendation, the whole blood should be collected in the dipotassium EDTA or a tripotassium EDTA. So if we talk about the cyclosporin assay traceability, the first generation was internal reference standard. If we talk about the second generation, this is standardized against the IFCC and other recommendation of the standardization of USP reference standard 1158-504. So this is also an evidence that our result is reliability and the accuracy is based on the traceability that what we have done for that. If we talk about the calibration interval, we have increased calibration the days from 7 days to 28 days in the second generation of cyclosporin. The limit of blank is 10 nanogram per ml as compared to 12.5. LOD and LOQ is 20 and 30, which shows that the between 30 to 2000 nanogram per ml cyclosporin values can be read without any dilution. If they want to run the dilution for greater than 2000, they can run this dilution by automatic or manual procedures, but you have to be review the package inserts guideline. If we talk about the onboard stability is a 4 weeks, and the two level of QCs are provided along with the free calibrators and the control, so the customer can monitor their low value results and the high value results from these quality control procedures. So if we talk about the cyclosporin first generation, what about sample preparation kit? Yes, in the end, I will share with you the package insert and I will explain you the package insert. Okay, Sergi, Rodigan, I will explain you, right? Thank you. So if we talk about the Siemens, they have also cyclosporin. And if we compare with the Roche, Siemens, and the Snipe, all we have a competitive principle for this cyclosporin analyte, and we have no firm therapeutic monitoring range for that. And the measuring range for the Siemens and the Roche, we have an improved range at the lower detection limit of 12.5, but they have a 30 value in same unit of nanogram per ml. If we talk about the limit of blank, they have a 20 and they have a 30 nanogram per ml, but we have a 12.5 nanogram per ml limit of blank. And with that, this is the first generation. So what we have done, we have talk about the sample volume. It's slightly higher than the other competitors, but in the next second generation, it's covered very promptly. So if we talk about the sample type, as you know that for the immunosuppressive drugs, EDTA whole blood is required for the treatment of the patient samples for the monitoring of the immunosuppressive drugs. So if we talk about the calibration frequency, the other competitors has 28 days or 7 days plus 28 days. We have an onboard reagent calibration frequency is 7 days. In the first generation, we will see about in the calibration frequency in the second generation as well. Stability on board is 1 month. They have a 56 days, and we have also a 4 weeks, a 1 day on board stability. So it is highly recommended by the SNIPE. You have to consume your reagent within the 4 weeks of the first installation. But if they don't have an option to utilize these 100 tests, the first option is they can order 50 test pack size. If they cannot order the 50 test pack size, they need to keep, according to the good laboratory practices, they need to keep the reagent on board when they want to run a patient sample, rather than to keep the reagent on board for a longer period to avoid any calibration curve failure for the next samples process. So if we talk about some more things, in a second generation, if we go to the second generation, you can see that we have an improved sample volume in terms of in cyclosporin, we have not improved. It's also a same like 80 microliter, and we have a whole blood sample volume. We have a measuring range between 30 to 2000, and then limit of blank detection and quantification is defined very aggressively. So if we talk about the calibration frequency, we have a 4 weeks calibration onboard frequency, and the stability on the reagent board is of onboard reagent is a 4 weeks. It means after the 30 days, it is recommended either to recalibrate the assay or you have to replace the reagent in terms of quality. If we talk about the test pack size, we have a 50 test and 100 test pack size. We have a QC and calibration includes in that, and then we can perform that. So if we compare the first generation of cyclosporin, so according to this data of commercially available cyclosporin SNIPE Meglumi, and using the clinical samples on the basis of 100 samples of these patients, they showed an excellent correlation between the lower range to up to the 2000 range value, and its regression value is 0.977, which shows that our assays is equally good as compared to the other commercially available cyclosporin assays. So let's talk about the solution from the SNIPE. We have all these three drugs monitoring like tacrolimus, cyclosporin, and the digoxin levels. While the Siemens will also have and the Roche have also offering all these analytes, including the Abbott. They are also providing these assays on their immunoassay platforms. While the Bachmann, they don't have a cyclosporin and the tacrolimus analytes from that platform. So if we talk about the other therapeutic drugs like amikacin, the digitoxin, the other drugs, the other competitors they have, we are in the process to focus on these therapeutic drugs. And if the customers need it, we can provide in the next phase because you know that according to the data as of today, we have 236 analytes available from that platform. So you are working also on a therapeutic drugs, which includes some amikacin, phenytoin, and then salicylate and the other analytes. So this is all about the immunosuppressive drugs and therapeutic drugs. So let's share with you some package insert, which is related to the therapeutic drugs and the pre-treatment steps. So let's share with you. You goes to the IFU. Hmm.
The following tutorial is buffer before using the.
Okay, let me check one more thing. Where is therapeutic drugs? Okay, let's go to the. Hmm. Have to check one more thing about the essay summary. If I have this, so let me check the immunosuppressive. Anemia. Okay, so let me share with you. I hope you received this protocol. This is SNIBE assay protocol units. So there's some conversion factors and they have some recommendation for the sample types, or you can see here that different sample types are for the different parameters are also given that. Like if we talk about here, you can see FK506, the whole blood sample type is recommended. And also for other drugs, they have some urine samples are recommended. In some cases, there are serum and plasma types is recommended. If we go further for this, you can see here in the rightmost side, if you go for inflammatory drugs monitoring markers like cyclosporin, if I go to again, that's drug. So you can see here, this is the therapeutic drugs monitoring and the immunosuppressive drugs. They have a linearity of like cyclosporin between 100 to 400 nanogram per ml, and they're at 12.5 to 2000 nanogram per ml, and they have a digoxin value of 0.9 to 2. These are the reference ranges we have established, but it is also recommended for the patient who are at the transplant situation or having some other therapeutic drugs monitoring they required. They have to ask with their physician. They will decide what is this a peak level samples or a trough level samples, and there are different reference range for the peak level and trough level for the different drugs. So that's why we have just give a generalized value for the reference ranges for the therapeutic and immunosuppressive drugs. But according to the organ transplant, the physician will decide what should be the reference range for such patient who have a kidney transplant, maybe the cyclosporin reference range or a therapeutic range is different from the kidney or a liver transplant. So this is depends upon the physician established reference ranges for that organ which they want to have some balanced value for that patient. So when you have this protocol sheet, that can be helpful if we go for some package inserts. Like if I talk about the if I talk about the SNIPE IFU, let me check with the SNIPE IFU. I have a one more package which contains all these SNIPE package insert in my hard drive. Let me check. I give you in a just next 2 or 3 minutes. Yes, I have got it. I can show you. Okay, so you can see here we have a package insert for all analytes. So if we go to the immunosuppressive drugs, any drugs which is required for that, we can go for that. So if we go to this tacrolimus, right? So you can see my screen. Please check. Okay, Antonke, thank you. So if you go to the assay's pack size, you can see on the rightmost side, the number of kit have a different pack size. We have available 100 tests and 50 tests. So this is the ordering reference number for this FK506. And you know that in this package insert, there's intended use is recommended. That's a quantitative determination of FK506 in human whole blood, right? So for this process, if you go further for some assay principles, you can see that here, this is a component shows that this reagent contains the magnetic microparticle bead, low high calibrator, that's a displacing reagent, buffer ABI, whole blood pretreatment reagent. So this is the whole blood pretreatment reagent. This is sodium lauryl sulfonate, and it's a 12.5 ml bottle for 50 tests and 100 tests. So they have also internal quality controls present. So how you can treat the samples, even with the Abbott, they have also a pre-treatment process for the immunosuppressive drugs. So this is the calibration process, and this is standardization of the internal reference substances show that this is the quality control, collection, and then separation is there recommended. And with the safety precaution, they have storage and test procedure. So first of all, for the first generation, this package insert belongs to the first generation. So you have to resuspension of the microparticle beads, and then the kit is loaded successfully. You have to ensure the microparticle beads are totally resuspended and homogenized prior to use. So this is the reagent handling procedures. If we go to the furthermore, the results calculation, interpretation, performance characteristic, they have also given that endogenous interface. And then, so in the package insert, the pre-treatment steps are not mentioned. They are provided with the separate procedures. So this is also they have in the reagent. There's some material which is required to treat with that. That's an whole blood pre-treatment reagent. There's the one procedures. So we can share with you the later about that. So let me check if I have an 1 minute so I can share with you. Otherwise, I can share with the later on in that. So for the tacrolimus pre-treatment. Okay, so in this package insert, they have a manual pre-treatment steps, but I have not right now. It takes a little bit time to search that part. If you have any question for that, I can share with, please share with me so we can respond. I can share with you about the pre-treatment steps of this cyclosporin and the tacrolimus as well. These are very simple steps when you perform at, even with the Roche and the Abbott, they have also a pre-treatment steps outside the instrument. Any other question if you have? Okay, so we have a next presentation, which is about the infectious assay. So we have can take 15 minutes break because it's a very long presentation. So it is better to take a 15 minutes break because in the next presentation, it's a little bit longer duration presentation, right? So let's go for a 15 minutes break. Okay, thank you. So, hello everyone. Let's start with the McLoomy Infectious Diseases panel. So, in this presentation, we will talk about the infectious parameters, especially for the blood donors like HBV, HCV, HIV, and the syphilis assays. So, as you know that, uh, let me check my screen is shared or not. Okay. Okay. So let's start with the infectious assays. So we have McLoomy hepatitis B surface antigen quantitative, surface antibodies, B quantitative, envelope antigen quantitative, envelope antibodies qualitative, and B core antibodies qualitative, core IgM qualitative antibodies, anti-HCV, HIV combo. All these assays has got a CE and List A certification. And as you know that for the world, if you want to work in a IVD market, if your product are CE mark, European certification and List A certification, that means that you can work or you can use these assays the worldwide except the USA, because the USA market need an FDA approval. So without FDA approval, you cannot run your IVD market analytes assays in the USA market. So let's talk about more about the infectious parameters. So in the first part, we will discuss about the hepatitis B virus. So what is the prevalence of hepatitis B virus in the world? The major burden of chronic hepatitis B infection, which is basically HBsAg positive by the worldwide WHO or Health Organization feedback, according to 2019 data, that this is basically 296 million people worldwide are affected by the hepatitis B. If we talk about the region-wise, the African regions, which is more prevalence of hepatitis B is 82 millions. And then is the Asia Pacific region, there's also 116 million people are living with hepatitis B. And then they have a Southeast Asia of 60 million. And then if we talk about the Mediterranean region, 18 million, and the European region has a 14 million with the American region of 6 million. So the estimatedly the global death with the due to this prevalence, high prevalence of 296 million hepatitis B chronic infection, there's estimated 820,000 deaths every year occurs due to this hepatitis B chronic infection. So you know the reason for this prevalence increases due to some gene mutation and then mutant sex. So if we talk about the HBV panel, you can see here the markers which we have a privilege to identify the chronic and acute and the recover phase of hepatitis B virus are the HBsAg, which shows an early HBV infection. Even those assays which can detect in the window period of hepatitis B infection, which are confirmed by the PCR, is an HBsAg marker. So the second marker is the anti-HBs. Its another name is hepatitis B surface antibodies. So these hepatitis B surface antibodies are protective antibodies after the vaccination or infection maybe occurs. So if the anti-HBs is also present and the HBsAg is negative, so it means that maybe the patient clinical history based on that maybe the patient in the past they have exposure or maybe have some vaccination for the HPSAG infection, so they are protected from that disease. If you talk about the envelope hepatitis B envelope antigen, so HBeAg is highly in infectious cases when the infection from initial stage to the prognosis stage is occurs. That's an it's called highly infectious. That's why envelope antigen is released when we see the structure of the virus. The envelope antibodies is shows that it's a weak ineffectivity. So the treatment, if they are patient is on treatment, the treatment is not has a significant impact on the patient prognosis. So the anti-HB is shows the early marker for the weak infectivity and that. So if we talk about the anti-HBC, they have a past or acute infection. And if we talk about the core IgM has an early infection marker for that. So if we see here the HBV infection, you can see here between 4 to 12 weeks is called a seroconversion type or the incubation period. And then after to the second weeks, you can see here the first elevated marker is a HPSAG. So the first marker which helps you to identify the infection with the acute infection is within the second week or the third month is an HBsAg, and the second marker is HBeAg, which is the envelope antigen. And when they have peak levels at mid of this acute infection weeks, and then it goes decline. And then there are other markers like anti-HBC that's contain core total antibodies is also the another name of anti-HBC, which includes IgG and IgM. And the other markers maybe is an core IgM, which is the individual which shows you the acute infection state, and then it helps you to identify either the patient is an acute phase or a chronic phase. It's another marker. The third marker is envelope antibodies, anti-HBE. This anti-HBE is increased in the acute phase, later acute phase, and then it remains in the early recovery phase, and it's sustained for the many years, or even after the recovery, it will remains in the bloodstream as an a marker for the history of past exposure. This is called an acute HBV infection with the recovery. But if we talk about the chronic HBV infection, you can see that there are the three markers which even last for the years and the months. The first marker is the hepatitis B surface antigen, and the second marker is anti-HBC, which includes IgM and IgM, and the third marker is anti-HBE, means hepatitis B envelope antibodies. So if anti is written, it means it's antibodies, or if in the last AG is written, it's mean antigen. So these are the markers which helps to diagnose the acute hepatitis B infection with the recovery and without recovery. And also the chronic HB infections can be ruled out with these markers. So these markers have a correlation with each other. On the basis of that, the chronic and recovered and the acute phase can be ruled out through this process. So if we talk about the diagnosis and the treatment and for the monitoring, they are according to some guidelines for the interpretation of these quantitative hepatitis B surface antigen testing. So as of today, there are two significant improvement. One is the HBsAg is called a quantitative. We call them QUAN quantitative, and another type is HBsAg qualitative, right? What is the difference between them? This is called HBsAg qual. So let me explain you what is HBsAg qual and what is the difference between HBsAg quantitative. So HBsAg qual is basically hepatitis B surface antigen screening assays, because these are the screening assays. And when you run the HBsAg qualitatives, you get the results either the cutoff value of 1, in some cases serum over cutoff index, S over COI index, If it is greater than 1, the result interpretation as a screening test should be like a reactive for hepatitis B surface antigen. If the patient value is less than 1.0 serum over cutoff I index, it's considered as a non-reactive for HbsAg that patient. So this is serum over cutoff index unit, which is a ratio, which is no quantitative, it's a screening assays. So if it this is that, this is considered as a negative and positive against hepatitis B subclinic. But in this terms of quantitative analysis, it's not only provide the interpretation of the results, either the reactive and non-reactive, and also this quantitative HbsAg testing can be used for the prognosis of the treatment. For example, if the patient is treated with the pegylated interferons for the treatment of hepatitis B surface antigen, when after 2 months, 3 months, or 1 month, according to the physician's requirement, when you order or physician order a testing for the hepatitis B surface antigen quantitative, they can reuse this results as a baseline results, right? So these results can be used as a baseline results, and after the treatment is started, and after, according to the physician requirement, 1 month, 2 months, or 3 months of the after treatment, when they go for a same HbsAg quantitative and the reference range for with the baseline value increase and decrease show the prognosis status of that patient against the hepatitis B surface antigen treatment station. So here is some algorithm according to the WHO guidelines, which is required for the hepatitis B surface antigen serological diagnosis for the routine, the population structures. So according to that, you can see that the HbsAg A1, these are there. They have HbsAg1 reactive report positive, HbsAg1 that's a non-reactive report reactive. So the compatible with the HBV infection, then they have no evidence of HBV infection, and they have active resulting immunization if it's ongoing risk for the exposure. So if you talk about the HbsAg, then the HbsAg is positive, then you go for HbsAg confirmatory, and then those reports positive, then maybe compatible with the HBV infection, proceed to the next testing or the quantitation of the viremia or a PCR is recommended. But in case of HbsAg, A1 qualitative is positive, HC A2 is negative, then it's go for inconclusive results. Further testing is appropriate is recommended. While for the HbsAg A1 non-reactive and the report reactive, no evidence of HBV infection is there. These are the guidelines of hepatitis B and C testing according to 2017 WHO guidelines, which are recommended. So if we talk about the structures of hepatitis B surface antigen virus. Just give me a 1 minute, please. I'll be back in just 1 minute, right? Thank you.
Okay, thank you. So if we talk about the structure of that virus, we can see that in hepatitis B surface antigen structures, it depends upon the outside. There is a HPSAG antigens which are residing on it. If you talk about the inside, there's a DNA-based virus, and this is enveloped with the envelope antigen, and then envelope antibodies are there. This area is called a core antigen, which is when they are replicated, it's released into the bloodstream, and their DNA contains also the polymerase, which helps to increase the value. So outside this core layer, there is a antigen that's called hepatitis B surface antigen. They have a middle protein of S plus pre-S2, and then you can see here they have a replication process in which they replicate into a S protein, M protein, and these S protein and N proteins when they have a release in the bloodstream, and this is the early marker for the infection when we are detecting the screening assay for the hepatitis B virus. So this is a DNA-based virus. If you talk about more about the HBsAg region, which has a main hydrophilic region, that's has an amino acids position of 121 to 147, which is defined as a determinant, which is common to all the HBV region. So this is the antigenic determinant side between the 121 to the 147 region. And then this is the determinant region is 124 to 147. If he goes to the into groups, they have some genetic types like ADW1, ADW2, and these further types are having different genotypes. So on the basis of this genotypes, different epidemiological distribution of hepatitis B common like type in Pakistan the type A and B are very common, and in the African countries type A, C, and G and H are very common, and in some other countries, like in Russia, the type C and D and E are very common types of hepatitis B genotypes, and then that. So it depends upon the epidemiological distribution of such viruses, genomic factors, and the different treatment for the different genomes. Some genomes are very tough, so the treatment procedure may extend up to the 1 years or maybe 8 months. In some genotypes, the treatments completed and the virus eradication occur within the 4 to 6 months of the therapy monitoring. So if we talk about the specificity of the assessment, you know that these are all the screening assays on the basis of clinical assessment for the specificity and the sensitivity. So according to the data, on the basis of blood donor, they have uses 5,101 samples, in which they have reported non-reactive 5,097. Only the four were reactive. If you talk about the hospitalized patient, they have run 205 patient tested, in which the 204 samples were non-reactive, only one were reactive. So this is the specificity we have. And if we talk about the interfering substances, they have 100 sample tested with non-reactive. There is zero you have with basically the non-specific reactions. So if we, on the basis of these different groups and the number of samples, we differentiate non-reactive and reactive. On the basis of their clinical assessment of their specificity, they have a 99.9% negative agreement. So there is a probability of in thousand, maybe the one samples may have a false negative probability. So if we talk about the HBsAg clinical assessment to the cross reaction, you can see that if we rent reacting substances or agent like anti-HCV, HAV, EBV, or other antibodies, including the CNCA and dialysis patient, HAMA antibodies, they have a non-reactive, and the reactive consideration is here. So again, we have a sensitivity and specificity very active within the acceptable range, and the performance-wise, we are best in class. So if we talk about the performance assessment on the basis of sensitivity, we have a undifferentiated samples type 89, we have a mutant samples type 63, we have a different genotype subtypes 259. So altogether, we have 411 samples which are reactive and on the different platforms from the mutant samples and differentiate types. So we have 100% sensitivity in terms of concentration, in terms of seroconversion panel. So we are more sensitive to the seroconversion panels in case. So if we talk about the performance assessment on the basis of the mutants, so do you know what is the reason why the hepatitis B surface antigen is spreading the globally? Any reason you have in your mind? So the batch hepatitis B infection is still spreading in the world, and even that the vaccine is also available for the hepatitis B. If we are talking about the C, if it is spreading, it's the realistic approach because the hepatitis C, there is no vaccine due to the seroconversion type of hepatitis C virus. So it's okay. But what about the B? We have a vaccine for the hepatitis B virus, but it's spreading. So the reason is the hepatitis B surface antigen mutants. So the mutants skip the detection of hepatitis B surface antigen infection or hepatitis B virus infection in human body, and that's why due to the limitation of the few mutants detection, that's why the scientist or this is one of the leading cause for the spread of that disease. But what we have on the basis of our internal performance assessment studies, we have a different panels for mutant panels for hepatitis B infection. So all these 10 panels, when we run from the SNIPE Maglumi platform of HBsAg quantitative assays, we have detected all these mutants of 10 mutants. So we have 100% sensitivity for the mutants as well. If we talk about the seroconversion panels, they have a different panels on the basis of 134 panels. We have a 137 panels detection of the seroconversion detection on the basis of 373 samples. So this is also a very big achievement between the Maglumi HBsAg quantitative in terms of sensitivity, in terms of specificity as well. So if we go further for other things like a method comparison of SNIPE, Maglumi, hepatitis B surface antigen, our first generation assay versus the commercially available hepatitis B surface antigen assay on the basis of a comprehensive Maglumi MBSA with the following correlation in terms of international unit per ml, we have a regression value of 0.9976%. So that data is based on number of 40 samples run in comparison with the commercially available first generation assay. So if we more compare with the other assays, there are a publication which shows helps them for regarding the Maglumi HBS AG assay, clinical utilization and different types. So in this part, we have some publication like according to this journal, the National Institute of Health in Lima, Peru, they have used the Maglumi quantitative HBsAg reagent to complete the analysis of hepatitis B virus in different region and age group in the Peru. This is the publication which shows that what they have done in this study of hepatitis B surface antigen for the analysis of hepatitis B prevalence in their countries. And the second publication is also belongs to the Union Hospital of Hangzhou, University of Science and Technology, and the International Joint Laboratory for the Infection and Immunity, the Eastern University Hospital of Germany, and Pritzker School of Medicine of the United States jointly published a publication that's show the AU value of hepatitis B combined with the X8 for the elimination of hepatitis B AAG prediction by the Maglumi. So this quantitative reagent was 0.80 and the AUC should be reached 0.97 when combined with the other reagent. So according to this publication, this is recommended that they can compare the Meglumi X8 versus the other competitors, and the results are comparable at the regression value 0.97. So if we talk about the comparison of HBsAg quantitative, this is the next generation assay we have. If we compare the SNIPE versus the other brand R, brand A, brand S, and brand D, we have a flexibility of 100 tests, including the 50 test pack size, while this brand R does not contain less than 100 pack size for that. So if we talk about the principles, they have a sandwich principle. They have a sandwich principle used across the board for the detection of HPSAG quantitative. If we talk about the sample type, yes, we have also a sample type of serum and plasma, which is equally recommended by the labs for that detection. So if we talk about the sample volume, it's a 100 microliter sample volume, and the other brand may have a 50 and 175 and 150 microliters, which is equally good as that. It's a minimal sample volume. So if we talk about the traceability, our HBsAg quantitative assay is standardized against the WHO third international standard 12 by 226, which is equally good as compared to the brand S, which has using the same international standard reference material for the traceability of Meglumi HBsAg quantitative. So in this calibration interval, we have a 14 days calibration interval as compared to the other of 7 days, 30 days, and 21 days and a 4 week, which shows that our assays is equally good in terms of calibration, in terms of traceability as well. So in HBsAg quantitative, what we have done, we have placed the 3 level of free of cost control. So this is a big difference among all the competitors, because the competitor has to purchase quality control material externally, and they need some additional cost for that 2 level of QC or a 3 level of QC. If we talk about the limit of blank, limit of detection, and the linearity, we are better than the as compared to the other competitors, but in some cases they have a lower good linearity of 0.02, which we have a 0.05, which has no clinical utilization for such low samples values. Any question? Okay. So if we go for a HBV panel, so this is very important interpretation with the HBV panel. As you know that we have a 5 major testing utilization for this, like one hepatitis B surface antigen. This is surface antibodies. This is envelope antigen. This is envelope antibodies, right? So envelope antibodies, and this is core antibodies. As you know that this core antibodies is the combination of hepatitis B core, IgG, and IgM, right? So this is combination of IgG, IgM, and in some cases IgA is also included in this core antibodies. So if the system shows you the results, surface antigen is negative, hepatitis B surface antibodies negative, envelope antigen are negative, envelope antibodies are negative, and core antibodies are also negative. There is no infection and there is no evidence of infection. Along with that, if the core antibodies are negative also, it means the patient is more susceptible to the infection because their immunization is not completed. So they have to go for a vaccination process if their core antibodies are low. And even after the vaccination of a booster dose of Ngrex B or some other pegylated interferons vaccine they have, and they have injected. So after especially the healthcare workers and the laboratory technologists and those people who have a chance of exposure in the diagnostic value. After 1 or 2 years or 3 years, they have to go for this hepatitis B core antibodies analyte, because this core antibodies analyte helps them to monitor their antibody titers. According to the guideline, if the hepatitis B core antibodies values are less than 100 in a vaccinated person, they have to go for a booster dose to keep their self protected against the hepatitis B surface antigen infection or hepatitis B infection. So if we talk about the next case, the surface antigen is negative, the surface antibody is negative, envelope antigen is negative, envelope antibody is negative. Only the core antibodies are positive. So you have to resolve the infection. You have resolved the infection that not immune to the hepatitis B virus. They have a resolving acute infection when the HBsAg disappear, but the HBsAg is not present. The maybe last option is they have a low chronic infection that HBV occurred, and this HBsAg negative but HBV DNA positive. It's a very low level. Most are also have an anti-HBC positive in these cases. So the third case for the interpretation of hepatitis B infection is the surface antigen is negative, surface antibody is negative, envelope antigen is negative, and the envelope antibodies and the core antibodies are positive. That shows that the resolving issue can be a weak infection, maybe that occurs, or some transition phase when the resolving of infection is going on. So if we talk about the vaccinating, if there is a surface antibodies are present, that's may lead to be monitoring for the vaccination status of the person. If we talk about the hepatitis B surface antibodies, hepatitis B surface antibodies is positive along with the envelope antibodies and core antibodies. That's to resolve the acute infection means. So the other case is the HBsAg is positive and the core antibodies are positive. So this is shows you the acute infection, and this is show the core antibodies are there. It means they may be a carrier. Currently, there is no infection, maybe some acute infection, maybe some cases, and they may be a carrier. So second option, these are to reflect that. So the further confirmatory testing is recommended in case of 6 case. If we talk about the 7 case, in this 7 case, you can see that the surface antigen is negative. Surface antibodies are positive along with the core antibodies are there. So it means that the infection is resolved and the immune to the hepatitis B virus. If we talk about the resolving infection, then it means that then further testing or the further clinical correlation is required. So if we talk about the eighth probability or interpretation of the hepatitis B infection, in this case, the HPS-AG result is positive, envelope antibodies is positive, envelope antigen is also positive. That shows the acute infection that's going to the resolve, or maybe a chronic infection, a weak infectivity is observed. So if we talk about the last interpretation of HBV infection along with the comprehensive hepatitis B virus menu, you can see here they have a hepatitis B surface antigen positive, they have a hepatitis envelope antigen is positive, and then core antibodies is positive. So this is shows that the HBV infection, it's a highly infectious case. Then though further treatment prior to the hepatologist recommendation, either they go for endoscopy or either they go for other invasive procedures to check the status of the patient condition, or they can go for a liver function testing as well. Any question in this case? Okay, if you do not have a question, let me take a one picture for that. Okay, thank you. I have to take one picture. Okay, thank you. So if we move forward for the other panels, so this is the comprehensive menu, which is provided by a few companies like Snipe, Roche, Abbott, and Siemens. I hope everyone knows that the Abbott has a leadership in the infectious diseases. They are the pioneer who provide the hepatitis B, C, HIV, and the other analytes detection kit in the IVD market. So now the SNIBE has also offering a comprehensive menu like hepatitis B surface antigen quantitative assay, anti-HBsAg quantitative assay, envelope antigen quantitative assay, anti-HB, anti-HBC core total, and core IgM as well. So the SNIPE assay has a comprehensive menu as compared to the Roche, Abbott, and Siemens, while the Bachmann, the missing part is envelope antigen quantitative is missing, and the core IgM is also missing in that part. The Biomerits and direct diasporin are also providing a very comprehensive menu for that. So we are again have a very comprehensive menu as compared to the competitors. If we talk about the hepatitis B surface antibodies, that's an quantitative analysis. In this quantitative analysis, which is grouped with the indication for the hepatitis B vaccination, all the kits are recommended for the hepatitis B vaccination, or unvaccinated children under that 19 years of age are also recommended for the vaccination. If we talk about the adult 19 to 59 years of age, and if we talk about the patient who have a 60 years of age and older than with the risk of hepatitis B, and the patient which have a 60 years of age and older without knowing risk may receive hepatitis B immunization or not. So this anti-HBs level of 10 MIU per ml or greater than have shown the strongly correlation with the protection against the hepatitis B virus. If this value is less than 10, it should consider as the surface antibodies are very low, so the patient is susceptible for the hepatitis B infection. So they need to be careful about that, or they may go for a vaccination or a booster dose if they are already vaccinated. So if we talk about the anti-HBs value, according to the approach to vaccine newspaper healthcare workers, so you can see here in this uh, days that the anti-HBs value is less than 10 MIU per ml. If this titer is obtained after 1 to 2 months of vaccine series, then you have to one dose HBV vaccine is required. Then you have to check either anti-HBs hepatitis B surface antibodies in 1 to 2 months. This anti-HBs, if again less than 10, then 2 doses of HBV vaccine is required. Then check anti-HBs in the 1 to 2 month. And then if it's again anti-HBs less than 10, not immune to the hepatitis B virus, then the doctor need to decide for the alternate vaccination process. So if this one dose HBV check and then it's a less than 10 hepatitis B surface antigen, they have to go for second anti-HBs testing after 1 to 2 months. And after 1 to 2 months, you can see that the value of this patient is greater than 10, which shows that it's immune to the hepatitis B virus. So this is very much important algorithm when you are handling different cases in your country for the infectious, especially the hepatitis B surface antigen issues. So if we talk about the envelope antigen quantitative, there is an excellent correlation between that sensitive and predictive seroconversion of hepatitis B virus DNA. And if we talk about change in the one treatment of hepatitis B virus DNA and hepatitis B serological markers, that shows that it's an excellent correlation between the hepatitis B surface antigen and the different weeks and then that. So the sensitivity and specificity is for envelope antigen in a correlation is excellent. Good. If we talk about the comparison of SNIB assay like HPV panel include the hepatitis B surface antibodies. So you know that the hepatitis B surface antibodies, these are a quantitative test. So if we talk about the panel for the hepatitis B surface antibodies, envelope antigen, and envelope antibodies. These are the quantitative testing from the Meglumi platforms. And if we talk about the qualitative testing, the anti-HBC and HBC IgM, they are a quantitative testing. So the two tests are a quantitative test like envelope antigen and surface antibody testings are the quantitative, like the envelope antibodies, core antibodies, and the core IgM are a qualitative test. So there is a one question here. If I would like to ask you, and I hope you will give me the answer. So my question is how we can check the IgG level. Okay, where is anti H B C I G G. Okay, if your customer want this test, what will be your answer? So I can give you in the chat box. If customer asks for anti HBCI IgG assay, what will be our response? Yes, we have, Antonke, we have reviewed the graph. If you want to go back, we can go back. So this was the graph, right? So your question was related to that, the when anti-HB after several days. So you can see that envelope antibodies. So you can see the envelope antibodies is here. This is the area when that's detected. It's between weeks to the months. You can see. So it's before the 3 months. It's an acute phase before the 3 months, and between the third weeks to the 8 to 9 weeks, it shows you the positive if anti-HBE is positive. So, but my question is related to the anti-HBE, right? If we go to there, I have a one question in this slide. If the customer asks, you are offering the assay like anti-HBC and HBC IgM, right? These both are antibodies, right? So where is anti-HBC IgG? Okay,
So we are not offering IgG individually. So what is the function? So first you have to IgM, you know, that helps you to diagnose the acute phase, while the IgM helps you to diagnose the chronic phase. So the core total and the IgM results helps you either this is the acute phase or chronic phase, because if the IgM value is higher than the IgM, sorry, core total bodies antibodies values are higher higher than the IgM. So it means that it's also include the IgG values. So they are a chronic case. If the IgM values and core are just close to each other, and that's because core total have an also a IgA, IgG, and IgM value, it means that's maybe some acute phase. So according to the description that's given, that can be used. Yes, total IgM. This is the total IgM for the core total. So if they want to calculate a theoretically, if they have a value of core total minus anti-HBC IgM, they may acquire a tentative IgG value for that, right? So if we talk about more about that assays, you can go for here. Let me share with you something more. Like we have at anti-HPS 100 test and 50 test pack size across the board. We have a 150 pack size kit for the HPV panel. They all are mostly they have a quantitative assays is based on the sandwich technique, while the competitive sandwich and the competitive and then the capture molecule base is by the hepatitis B antibodies, core antibodies, and core IgM antibodies. If we talk about the sample, we have the minimum sample volume of 40 microliter, 50 microliter, 20 microliter, and 40 and 10 microliter. If we talk about the traceability, we have a WHO second international standard standardization for that. We have an international standard for that. For envelope antigen is WHO international standard 129097/12. If we talk about the qualitative analysis of hepatitis B antibodies, we have a WHO standardization of 129095/12, and the WHO internal standard for the anti-HPE is the traceability for that assay performance and the reliability. If we talk about the qualitative analysis of anti-HBC, we have this assay standardization against 95/522 material for that. If we talk about the core IgM antibodies testing, this is a reference standard that the Paul Ehrlich Institute of Germany, we have a traceability for that. So in terms of calibration interval, we have a 2 weeks calibration stability recommendation in case of anti-HBS, BAG, anti-HBC, and HBC IgM. But in case of envelope antibodies case, we have a onboard stability of calibration is 28 days. So if we talk about the internal quality controls, we have a 3 level of QC in surface antibodies case. In case of envelope antigen, we have 2 levels. But for the qualitative analysis, we have a negative and positive value of 2 levels, which are free of cost along with the calibrators in these assays from the hepatitis B virus panel. We have for the quantitative assay, we have a panel for the limit of black and detection, and on the basis of which we have a linearity of 0.5 to 1000 MIU per ml in case of anti-HPS, and if case of envelope antigen case, this is between 0.1 to 200 international unit per ml. So the cutoff for the entire HBS is 10, and if we talk about the cutoff for envelope antigen is 0.1 IU per ml. So if the results higher than 0.1 IU per ml in hepatitis B envelope antigen are considered as a positive for envelope antigen. So this is a result interpretation because due to these are screening tests, and the ratio usually used by the lab either in terms of IU per ml or serum over cutoff index, they can use that for a reference rate for that. So if you go for a further, the different panels that we are offering from the proficiency testing. We have a proficiency testing like recast, that the recast cycles of March 2023. The results are shows that the non-reactive negative result, then N value is less than 5. That's the non-reactive. There is no one has sent that the intermediate or reactive or positive samples. The number of labs is the one labs, and they have a non-predictive values for that. So this is the 100% non-reactive confirmation from the third-party assessment. They have a Roche. They have also a non-reactive. This sample is also a non-reactive. The participation is 19. The Abbott Architect number of participation is 16. The Siemens Atlica instrument have participation number of 10. Number of participants are thus 10 for the hepatitis B core antibodies assay. So the different companies had a different participation, including the SNI Meglumi hepatitis B panel. So if we talk about the hepatitis C virus, as you know that this is an also in a hepatitis around the world, there are 71 million people globally are infected with the hepatitis C virus. So this viral hepatitis C virus distribution globally is significantly high in the countries where have an Africo-American countries, and then those countries where we have some prevalence of this in the Mediterranean area. Like if we talk about the Western Pacific region, we have a 14 million people are infected with hepatitis C virus. If we talk about the Southeast Asia, the 10 million, which is equal to the African of 10 millions, and the European has a 14 million hepatitis C infection as compared to the 15 million of Mediterranean, Eastern Mediterranean region. And this African American countries are just 7 million people are infected with the hepatitis C virus. So the estimated number of acute hepatitis C virus from 2011 to 2019, they are increasing gradually. And this estimated number of infections from between the 2011 to 2019 is between 17,100 to 57,500 people are infected with the hepatitis C virus. So if we go for a serological pattern for the HCV infection, so you know that we do not have an antigen testing for hepatitis C virus. We have antibodies testing for the hepatitis C virus. So if we talk about the time exposure, that's in a window period called the window period in terms of months and the years, and then the concentration is increasing how this way. So the antibodies are from the 0 day start increasing gradually, but in the second after the second week of after the second month of the exposure, it gives you the symptoms and it moves very aggressively. So for the PCR or nucleic acid testing, it is recommended to go after 4 months. After the 4 month exposure, then they can go for a HCV-RNA detection for the confirmation of hepatitis C infection. So the first marker is the antibodies. As of today for the HCV, it helps to differentiate that, and along with that, the biochemistry markers also increase within the 1.5 month of the exposure, and then it shows you a symptoms with the same time frame for when the HCV RNA can be also be detected. So this is important for the serological testing is the window period. So what is the window period? If I ask you about the window period, basically the window period is that period in which the virus are replicating and increasing their numbers, and the diversity market, if the assay is detected by the or the parameter is detected by the analyte is detected by the distributors, the manufacturers are considered to be the most sensitive and specific kit for that. So this is HCV antibodies titer, different values, and then they following by the HCV acquisition, and you can see that in some assays they have detected on the 16th day of the exposure from the 0 day for that testing. So average after the exposure, if there's some discrepancy occurs, so it is highly recommended to write on in the reporting format that this is a screening test. Advised to recheck after 4 to 6 weeks in case of clinical correlation is occurred. So if someone has given a positive or someone has given negative, or you are reading that sample as a positive sample, you need to check that patient after 4 to 6 weeks on a both analyzers, and then you can conclude that if the competitors systems gives you the positive results, so the window period detection limit for that competitor is less than the SNIPE. So SNIPE is more sensitive and specific in terms of detection in the window period as well. So if we go for the CDC guidelines for initial hepatitis C antibodies positive case. So you can see here, if the HCV infection is negative, its interpretation as the non-reactive, there is no HCV antibodies detected, so the no probability of HCV infection is occurs. If the antibodies are positive, then they have to go for a HCV RNA test, either for the nucleic acid testing or the PCR testing. If the new RNA is detected, then go for a current HCV infection and then link for the care or the treatment. If the antibodies are positive, but the HCV RNA is not detected, so the no current HCV infection, additional testing or appropriate are recommended, because this HCV results is discrepant results. If you have found the patient with other competition having a positive, negative results, or for anti-HCV, and you are generating a positive results for the HCV antibody testing, then you have to go for after 4 to 6 weeks for clinical correlation. I have to draw the samples and run on the both analyzers to identify which has detected in early during the window period, and which one has picked the false positive, or what is the dangerous, either the false positive or false negative. In IVD market, this is a double-edged sword. False negative is also dangerous as compared to the false positive. But in the blood screening assays, false negative is the dangerous as compared to the false positive. So this is the screening algorithm for the HCV infection. If we go for the structure of this virus, so the HCV viruses are RNA-based viruses, and you know that this RNA-based viruses has some capsid protein, that's in the core protein. Outside that, then they have a E1 and E2. They have a lipid bilayers, and then they have a HCV genomic RNA containing the virus RNA and the other strings. So if we talk about the structure of this virus, when we open that structure, this can be summarized into a structural protein and non-structural protein. So the structural protein are from the core, that's the nucleocapsids, and then we have a glycoprotein envelope, that's E1 and E2, and they have an ionic channel of P7, and the cysteine protease enzyme, which is NS2. So for the non-structural protein, they have NS3, that's an serine protease RNA helicoase, and then NS4A, NS4B, that contains the NS3 core factors and the membrane web formation, and then NS5 is an phosphoprotein protein particles of protein structures. And if we talk about the NS5, that's an RNA-dependent RNA polymerase types. So from 5' to 3' DNA structure shows you the different structural protein and the non-structural protein for that. If we talk about the hepatitis C virus genotype, specific antibodies response against the recombinant HCV protein, that's according to that, their core have a 95% responsibility. And then they have detecting NS3, NS4, NS4AB, NS5A, and NS5B. So the percentage wise of the positive area for the A number of positive area, you can see that the core has a 97%, the E1 has a 22%, E2 has 31%. The NS3, NS4, and NS4B, NSA, and NSB, they have a different percentage of positive serum as compared to the antibodies titer of that patient. So again, if we talk about the generation of the anti-HCV, so as you know that there are some principle for the generation. The one is the internal generation, like for us, like prolactin first generation, prolactin second generation. So there is an internal improvement, either in terms of sample volume, in terms of quality control solution, in terms of reference ranges, in terms of pre-treatment steps, in terms of any reagent improvement. We call them internal generation. But the externally, according to the IFCC guidelines or the some other regulatory authorities guidelines, there are some criteria for the generation of each kit. Like if we talk about the anti-HCV kit, there is a generation concept about that. So let's see about the concept. So here you can see the HCV structure is divided into two parts. One is the structural protein, the other part is a non-structural protein. So if we talk about the protein, there is a core, E1 and E2 are the structural protein, while NS2, NS3, NS4B, NS5BA, NS5B, these are the non-structural protein. If we talk about the function, that's the core antibodies play a very vital roles for the preparation of the nucleocapsids, and E1, E2 are for the envelope functions. And if we talk about the non-structural protein, they have NS3, NS2 has an protease activities, then the NS3 has an serine protease, and then the helicase antiprotease, and the NS4A has an NS3 cofactors. Along with that, the phosphoprotein and RNA dependent and polymerase reactions are also on that. For the first generation kits, it was proven that the HCV, anti-HCV kit, if they detect the NS4A and NS4B analyte it, so they can be considered as the first generation. Along with the NS4A and NS4B, if they detect the NS3 along with the core antibodies, so that can be considered as a second generation. For the third generation, core along with the NS3, NS4A, NS4B, NS5A, and NS5B, if it's detected, is considered as a third generation assay of megalome anti-HCV. So these are the generation for the anti-HCV. Do you have any question? Okay. So I have a one question. I'm writing in the chat box, right? So today, as of today, in a anti-HCV case, we are using the third generation. Okay. So if if in the IVD market, if they launch HCV combo, if they launch HCV combo, what will be the generation and what this it detect? So these are my three questions. So you can write your answer in the meeting chat, so I can make a clear concept about the generation. Okay, I'm waiting for your response. When you try or give me the response, then I will give my answer for that. You can think and then the generation, more protein detect, maybe. But when you keep the generation to detect the more protein, it's a sensitivity will be compromised, you know, because the sensitivity and specificity are reciprocal of each other, have an inversely relation. If you increase the sensitivity of any parameters, then the specificity will be compromised. If you increase the specificity of any analyte, then the sensitivity will be compromised. So if you usually detect, yes, Roman, you are perfect. Excellent, Roman. Your response that the combo means antibodies and antigen. If this combo is detected, it means it will be hopefully the fourth generation kit. for HCV, right? For hopefully, right? And it will detect both HCV antigen and antibodies. So that was my answer. So the maybe a fourth generation of HCV will detect both antigen and antibodies, right? And the generation depends upon the sensitivity, specificity, and the detection capability according to the IFCC guidelines or other authorities which provide the traceability and accountability for the sensitivity and specificity. So let's compare the Meglumi anti-HCV assay with the other competitors. So, like if we talk about the SNIPE, sorry. So if we talk about the SNIPE anti-HCV, we have comparison with the R, A, N, S, and B competitors. So if we compare the SNIPE anti-HCV kit can detect score, NS3, NS4, and NS5. So as compared to the other competitor, only the S competitor has an capability to detect core NS3, NS4, and NS5. With comparing other brand, like the other brand, they are missing NS5 because when you have increased the NS5 detection in your assay, the probability of false positive both, but the possibility of false negative is almost zero, right? So this is the features where while we have in a SNIPE and one of the other competitor, while these two competitors are missing part is NS5. So this is the edge of NS5 and the disadvantages as well as for that. So if we talk about, because in the IVD market or screening market, false positive is okay, false negative is dangerous. So false negative is not acceptable. False positive, because this is a screening assay, you can go for a further confirmatory testing, and these further confirmatory testing explain the customer to detect the true possibility of that first initial screening assays positive results. So if we compare the SNIPE versus the other competitors in the market, you can see that we have a 50 test kit, 100 test kits with the 200 test competitors with the 5,000 tests and the 2,000 pack size. So they have a different pack size for the anti-HCV. If we talk about the principle, all they have a sandwich principle except the S brand, they have an indirect sandwich principle. So if we talk about the sample type, serum and plasma, both are recommended across the board. If we talk about the sample volume, we have an equally good competitive sample volume as compared to the other competitors. If we talk about the onboard stability, 1 month is the onboard stability as compared to the 7 days and the 15 days, and this as competitor has a just a 41 days calibration stability. The cutoff value in terms of arbitrary unit per ml is 1, and the borderline is between 0.9 to 1.0, and the serum over cutoff index in Abbott is 1.0. And for the S SNIPE has a 0.8 to 1.0, that's an equivocal region or a borderline case. If the value fall between 0.8 to 1.0 serum over cutoff index, it should be considered as a borderline reactive cases, or they need to be advised to recheck after 4 to 6 weeks. That's an important part. So again, we have a free of cost calibrators controls, but from the competitor R, A, and S, they need a calibration, including that that's an include the free of cost QC and calibrators are available across the board. So if we talk about more, we can go for a sensitivity assessment. We have a different bleeding samples from the different donors, and we have check on the first day of a bleed and the 0 day and the fifth day, 7 days, 13 days, 16 days, 17, 20 days, 26, 28, 33, and 35 days. on the different members, they have a different bleeding times, and on the basis of the SNIPE results, shows that we are anti-HCV detects the HCV infection earlier. So we have detect at the seventh day and the 13 days of the bleeding time. Only the brand A has detect on the 13 days, while nobody has detect at the 7 days. So we have an edge of early detection during the window period. It's also and gives the sensitivity assessment that our sensitivity is 100%. So if we talk about the third party controls, the SNIPE range is negative is less than 5, the other brand has less than 1 and less than 1. If there's 0.1 NCU value, we have a 13.8, they have a 26.7, and the one is a 131 value on the SNIPE third party controls assays. If we talk about the sample type, undifferentiated sample type 274 were reactive, no one non-reactive is reported. If we talk about the different genotype from 1 to 6, 126 detected non-reactive, no one is undetected in this situation. If we talk about the total samples, there are 400 samples were detected on the basis of undifferentiated and the differential diagnosis type. Their non-reactive samples are 0. So the SNIPE and anti-HCV shows a superior sensitivity in terms of the detecting the window period and the third party QC and the undifferentiated and different genotypes for the case of anti-HCV detection. So if we talk about the specificity, we have a specificity assessment in terms of blood donors and the number of sample tested for the non-reactive and the reactive cases. So if we talk about the blood donor, when we have studied the blood donor, that's in 543, and the reactivity is reactives are 10. So if we talk about the hospitalized patient, they are 22 and non-reactive, only the 0 are the reactive in interfering substances, 129, 128, only the one samples is reactive with the interfering substance. So the negative agreement or NPV, that's a negative predictive value is 99.8%. So across the cross reactant, they have a different autoimmune disease substances or agent like hyper IgG the pregnant woman, dialysis patient, rheumatoid factor positive, syphilis positive, anti-HIV positive, EBV antibodies is positive. All these varicella zoster virus positive, P simplex virus, hepatitis A virus positive antibodies, surface and core antibodies are positive, envelope. Different cross reactants also detected on that, and then they all were non-reactive with this, and then the reactive was also no discrepancies as reported for that. So again, we have a sensitivity and a specificity in the best in the class. It's a 99.8%. So do you have any concept for sensitivity and specificity? It's clear. Okay. If it's clear, I will go the next slides, right? So on the basis of different panels, these are the seroconversion panels. We have a comparison with the other competitors. We have compared with the SNIPE. We have detected different sero panels at different stages, and then we have a conclusion of positive results of 70 out of 205 and 63 as compared to the other competitors. We have a false positive 0, and they have a 22 false positive probability. That's the most probably in Abbott. So you will see in our assays, the probability of false positive is very low as compared to the other competitor A, which has a 22 out of 42 samples. They have a false positive, and they have also missed one sample out of 205. They have missed some such samples of as compared to us. We have a 70 panels being detected during this. So this is specificity assessment on the basis of different seroconversion panel shows that our assay has a maximum seroconversion sensitivity in this panel. So if we talk about the certificate, according to INC certificate from the Brazil, has been published. That's for the SNIDE that the assay performance meets equally good according to the other competitors in the market. So if we talk about the third-party assessment for the accuracy and the reliability, we have an example for the EQAS, that's an external quality assurance program from the ECAS company. They have detected that your results is negative, that's a less than 5, and then the negative results is 100%. Intermediate, no one has given, then no one has given the reactive and positive. This is a good agreement with other manufacturers as compared to the others like the other competitors like I 2000, they have a 99 participant and they have the presence of non and 1% of the reactive case. And in the LTI, they have a participation of 84 samples and they have a non-reactive 100% is non-reactive, no one is reactive reported in this case. If we talk about the architect I 1000, they have a 75 instrument has been reported 100% non-reactive, and the electrochemical Roche Cobalt has an 100%, BioRed Mona Lisa plus 100%, and then the other Cobas E411 and the BioRed Mona Lisa Ultra has an agreement of non-reactive is 100%. So this is shows that the accuracy of the assay performance meet the expectations of the customers in the NTSCV case. So if we talk about the publication, we have a publication. So we have a publication. If we talk about this publication, according to the National Institute of Health in Peru, according to that, they have used the Maglumi anti-HCV reagent to analyze the presence of HCV in the different region and age group in Peru, and then they have collected data for that. If we talk about the Genius Laboratory of Trinta, Albania, they have used the Maglumi anti-HCV reagent to evaluate the performance of HCV first generation and second generation, third generation reagent, and they have shown that the anti-HCV reagent is more sensitive as a third generation assay. So this is a conclusion that our third generation assay is the more sensitive and reliable in terms of sensitivity, specificity, and the results accuracy in terms of third party reagent. So if we want to start a syphilis, we can start right now. This is the third part, which shows you the syphilis part, and then we have a syphilis. So let's start with the syphilis. The prevalence of syphilis around the world, according to 2016 data, incidence with the chlamydia, gonorrhea, and trichomoniasis, and the syphilis, these are the STDs called sexually transmitted diseases, according to the WHO region of the America, region of the Africa, Europe, and then there are around in the countries like 75 million people are impacted with this STD diseases. And then the African region, there's 86 million people are in 86 million people are exposed to these STD diseases. And in this Asia Pacific region, 34 million, and then the region which has an like China, 108 million people are impacted, while in the Russia there are 23 million people are impacted with that. So according to this WHO global region and the incidence cases for the sexually transmitted incidences from 2016 estimated that there are new cases of these 4 STD in 20 are shown at the bottom right of the figures, and you can see that. So according to the 2023 data for the WHO estimated that 377 million people new cases are reported for that STD diseases like the chlamydia 129 new cases, gonorrhea H2 cases, syphilis 7.1 million new cases are reported with the trichomoniasis of 156 million new cases. So the prevalence or the prognosis of the incidence increased cases in the last 8 or 9 years is increasing more. And according to that, we have to know about the prevalence of that diseases. So let's go for a background. This is about the bacteria which cause the syphilis. So the bacteria name is Treponema pallidum. Subspecies is the pallidum. This is the genus. This is the genus. That's the Treponema. So this is a chronic systematic infection which is caused by this virus, and the structure of this virus overall, sorry, it's a bacteria. The overall length is 10 to 20 microsiemens, and this diameter is 100 to 200 nanometers. So this part is the flagella motillar, and this is the tip structure. This is the cytoplasmic filament, and this is the cytoplasmic membrane, and this is the outer membrane. If we see the structure of that, they have the different gene like the glucose and that. This is the TPK84, TPK319, TPK 171, TPK 435, and TPK 574. So this is the genomic structure and the phenotypic structure of that virus. This syphilis is include cause the infection into three different stages. The first stage is called a primary stage. The secondary stage, the latent stage, and the tertiary stage. So this helical chronic systematic infection of the syphilis has a TP15, TP17, and then TP47. These are the extracorporeal lipoprotein of the syphilis. And these large number of studies show that the TP15 and 17 and TP47 are not only have as high immunogenicity, but then can also be used as a diagnostic antigen for the Treponema pallidum. So this TP15, 17, and 47 are used as an diagnostic tool for rule outing the Treponema pallidum infection. So if we go to the serological course on the basis of time of infection and the weeks, in the between the first and the second week, the treponemal IgG bodies are increases, IgM antibodies are increases. This stage from the primary to the secondary stage is considered as, and at the end of the secondary stage or at the beginning of the latent stage, the IgM values decreases. So if we talk about the other markers like IgG and then non-treponemal IgG and IgM, these both are increases at the third week of the infection, and then started at the secondary stage to the latent stage, and then IgG antibodies stage at that remains is that. While the non-treponemal that the treated IgG and IgM remains longer period. So this is basically the infections start, and then it remains in a weeks, and then it's prolonged for up to the 20 years of infection that can be had. So this is the different stages that the primary, secondary, the latent, and the tertiary stage of this syphilis serological course shows that it remains in the longer duration. So you can see here the serological detection, the common methods that the non-troponemal assays, like syphilis infection, that lead to the production of non-specific antibodies that can also react with the cardiolipin. So this non-treponemal assays, serological values for that, that's considered as that. So the VDRL, the Viral Disease Research Laboratory Test, or the Rapid Plasma Region RPR test, and the trust tolerine red unheated serum test, or USR, the unheated serum regain test, are useful for the non-treponemal assays. For the treponemal assays, look for the antibodies that are direct results of the infection, and the anti-treponemins IgG, IgM, and known lesser degrees of IgA. So the fluorescence treponemal FTAB, TPPPA, that's the treponema pallidum particle agglutination assays, and the EIA, chemiluminescence assays, and the microbeads immunoassays are the tools for the treponemal assays detection in serological methods detections. So these are the sensitivity of different methods in terms of percentage wise. If we divide these method on the basis of their primary, secondary, latent, and tertiary sensitivity wise, you can see that for the primary and secondary phase, the dark field microscopy is more reliable, but they have no clinical significance in terms of sensitivity for the latent and the tertiary stage. If we talk about the PCR, this is one of the most better sensitive technique for all stage, either the primary, secondary, latent, tertiary, or the specificity in terms of specificity. The PCR is the most sensitive and the reliable technique. But due to the cost factor, if the serological testings are available, then they have to go for some other testing. Like chemiluminescence, you can see that the chemiluminescence has in a primary case, they have a 98% sensitivity. For the secondary, latent, and the tertiary stage, they have 100% sensitivity. If we talk about the specificity, then the specificity is 99%, while for the confirmation of this chemiluminescence automatic technique, they can be either technique with the VDRL, RPR, the Western blot, TPPHA, or ELISA techniques can help them. So in the next slide, you will see about the sensitivity of different method and the algorithm as well. So for the traditional algorithm for the syphilis screening is that on your left side, first you have to go for the RPR screening test. Once you have done the RPR test, if it's positive, then go for a treponemal test. The syphilis is positive, then go for a treatment. And if syphilis, the treponemal test is negative, then the negative for the syphilis. And if the treponemal screening test is negative, that should be considered as a negative for the syphilis. If the treponemal screening test is positive, then go for RPR. This is the one-step go back procedure when the chemiluminescence technique is introduced. If the RPR is negative, then the TPPA is go for that, and if the TPA negative, then you have to consider as a negative syphilis. So on the basis of this SNIPE assay positive test, then this positive RPR is go for a syphilis. If this TPA is also positive, then he is considered as a syphilis on the historical background related. So this is the reverse algorithm for that. And these are the specific specification and the comparison between the SNIPE first generation and second generation versus the other Roche, Abbott, and Siemens assays. So we have a edge in the test spec size. The sandwich technique is same. We have a minimum sample volume. We have a serum and plasma recommendation with the 2 level of QC free of cost. We have a 14 days calibration interval with the cutoff value of 1.0 as compared to the one of cutoff value of 1 with the Roche and Abbott. They have a C over I index ratio, and we have a fragment detection of treponemal palladium N15, N17, and N47 as well, which has the this Siemens has an detection value. Yes. So this is about the specification and comparison of SNIPE syphilis assay, first generation, second generation versus the other competitor in the market. If you have any question for that, you can ask me. So if we compare what is inside that, so we have an edge of detecting TP15, TP17, and the TP47 in our SNIPE assay. So the Roche and Abbott has a capability to detect all these three fragments for 100% sensitivity and specificity, while this has a missing of TP47, so the probability of false negative in the Siemens group is more as compared to the Roche and Abbott. So we are state of the art assay providing from the SNIPE platform as well. So if we compare the 100% of negative predictive value and the positive predictive value, our assay tested on 534 patient samples. With the positive samples, we have a zero negative or reported on that. So the total 534 is reported. If we talk about the syphilis negative, they were confirmed 745 samples. We have detected 745 negative samples. If we talk about 534 and 745 samples, they were total altogether 1279 samples were tested, and on the basis of this, the sensitivity is 100% on the basis of 95 percentile, that's a 99.91% about, and the specificity is 100%, which is 99.92% versus the 100%. So this is about the sensitivity of and specificity, or you can say a negative predictive value and the productive predictive value for these assays. So if we talk about the sensitivity assessment on the basis of different panels, seroconversion panel, we have detected, and we are detecting early at the day 31. The Maglumi is detecting all the panels which are available from that. The other competitors like Brand T and Brand T, they were not able to detect that results on day 31. So we are during this seroconversion type, early details, we have a superior sensitivity in terms of early detection. If we talk about the specificity assessment and the sensitivity interferences, we have seen that potential cross reactants that can interfere in the negative results, SNIPE positive results, they are not impacted with these potential reactants. And we have also have a study of the interferencing substances like cyclosporin, levodopa, any therapeutic and immunosuppressive drugs up to this concentration. So the overall Maglumi shows a superior specificity even in the presence of some reactants or a potential cross-reactant substances present in the patient serums. We have a highly specificity in this term. If talk about the third party assessment, we have a participation for the qualitative detection of this syphilis and Chigas. We have a good agreement with the manufacturers about the participation of that. The other participants have also a non-reactive and reactive participation, having a less than 100% in some cases of Architect I-2000 case, and in also with the but LND cases, they have a 19 participation, but the reactive positive confirmation is just a 95%, and we have our results positive confirmation is 100%. So we have a very good agreement with the manufacturers in terms of accuracy. If we talk about the HIV prevalence is more in the African countries as compared to the other countries, and then the more than 2.5% prevalence is in Russian and in the North American countries like Brazil, Peru, and then the other countries. If we talk about the structure of the HIV structure is having like this RNA-based virus, and they have a different type 1 than the lipid bilateral gear, GP4, and they have 124 nanometers and roughly spherical in shape. Their outer lipid membrane envelope contains GP41, GP120, and the HIV type 2 contains GP36. They have a spherical substrate like protein P17 and P24 protein formation of half conical shell capsids. And then they have a virus RNA genome, enzyme, and other component from the host cells. So this is about the virus background of the structure of that virus, of HIV virus. So the background, there are two main types of HIV virus, HIV-1 and HIV-2. The most common type is HIV-1, and the HIV-1 has also further classified to subtypes. That's an M major group and is responsible for the majority of the HIV epidemic. So if we talk about the M group, the M group has an further genotypes known as an A HIV-1. There are subtypes A, B, C, D, E, F, G, H, I, J, and K as well. So the HIV group 1 is the strain of the HIV is responsible for the global HIV epidemic. This is HIV-1, group M, N, O, P, and the M has a further A, B2 up to K and CRF types of further subgenotypes. So you can see that the global distribution of HIV on the basis of that, but the major part is distributed, which is leading to the epidemic for the HIV is HIV M type. So this is serological course. The interesting thing about the HIV infection is basically window period. So in viruses, HIV have a shortest window period, which is 7 days. So you can see that after the 14 days, in case of discrepant results, you can conclude the patient value of with the fresh sample after 2 weeks recheck with the fresh sample gives you the conclusion in terms of RNA base or the DNA base or a serological testing base. So you can see here the HIV infection, if you consider as a 0 day, on the day 11 or a 10, you can see that the HIV RNA can be detected in the bloodstream, or HIV-1 or P24 antigen is also present in that stream. After 20 days or 22 days, 3 to 4 weeks, the antibodies are also start to be available in the bloodstream, so that can be detected. So the goal for the infectious assay is that to detect during the window period, and this window period is this. In this window period, which companies detect more early, that can be considered as the more reliable in terms of sensitivity and reliability. So this timeline for the HIV infection response and the window period for the test is 0 days. That's an actual infection. Between 7 to 4 days, viral load or a PCR is the most reliable technique to detect in the 95% within the 3 days of the infection. And if we talk about the day 16, the P24 antigen is also developed in the majority patient between 1 to the 8 week. The seroconversion type is 70% people detect their symptoms, get their symptoms, and that can be detected in the 4 weeks. So after the actual infection, after the 4 weeks, the seroconversion type is detected, and then the day 28, some people have an antibodies positive using the fourth generation test can be detected that. For the day 19, that there are people with HIV infection tested positive by the third month. So the virological and serological marker concentration during the HIV infection helps them to diagnose early during the seroconversion is the best goal for the HIV infection. So if we talk about the generation of HIV kits, you can see here the first generation kit was launched in 1985 that was detecting only the HIV-1 IgG antibodies. In 1989, the scientist or the diagnostic field biologist has detected the second generation kit have developed that the second generation which detect HIV-1 and 2 IgG antibodies. In 1992, they have introduced the third generation kit, which can detect HIV-1, including group O and HIV-2, IgG and IgM antibodies as well. In 1998, we have detected the scientist has established the fourth generation kit, which detect the HIV-1, including group 0 and IgG and IgM for group 2 plus P24 antigen. That's why we call them a fourth generation assay. So maybe in next couple of years, they have launched the fifth generation, which includes the HIV-1, HIV-2, all subtypes, including the P24 antigen. So that may call them a fifth generation HIV kit. So this is the CDC guidelines for the algorithm of HIV test. If the patient has the HIV negative. So that could be considered as HIV-1 and 2 antibodies negative and P24 antigen negative. If the patient has a positive initial HIV combo 4th generation assay, then the HIV-1 and 2 antibodies differentiation test is recommended. So there are there are four possibilities. One may be HIV-1 positive, so the HIV-1 in antibodies detected, so the probability of HIV-1 infection is there. If the two antibodies are positive, so the HIV-2 antibodies detected, it means HIV-2 infection is probability is more. If HIV-1 and 2 antibodies both are positive, so the probability of HIV-1 and 2 infection is more. So if the HIV-1 is negative or indeterminate, HIV-2 is negative, then it's time to go for a further confirmatory test through the nucleic acid testing. So the NET testing or PCR for the HIV-1 is recommended for the HIV-1 if then the NET testing is positive, so the acute HIV-1 infection is there. If the HIV-1 NET testing is negative, then it should be considered as a negative for HIV-1. So this is the algorithm according to the CDC guideline for the HIV infection when the 4th generation antigen antibodies assay is positive. So this is the kit into detail about the HIV. We have a 50 test and 100 test combo kit available, which have a sandwiches, qualitative analysis. The serum and plasma both are recommended. Sample volume is 200 microliter. The cutoff value is 1.0 arbitrary unit per ml. The analytical sensitivity is 0.7695 IU per ml, which shows that this is the standardization against the NIBSC code 90 by 636. We have an analytical specificity of 100%, and we are detecting HIV-1, GP41, HIV-2, GP36, and P24 antigen. We have a 4 level of QC free of cost available along with the reagent. This is shows you the 4 level of QC. What does it mean? So if I ask you the one question, why SNIPE gives 4 level of QC in HIV combo. So, sorry. So what is sensitivity and specificity? Okay. I will tell you at the end of the session. Just give me the answer. Why SNIPE gives 4 level of QC in HIV combo kit? Usually we have listened that there are 2 levels, low level and high levels. Maybe in some cases 3 levels, low level, medium level, and high level. Why SNIPE offer 4 level of QC? And what are these 4 level? If you have any idea, you can discuss and give me the answer. It's a very typical question and a very brainstorming question. You will learn a lot about that, right? So let's discuss within your team and give me the answer. One person can give the answer. I will be happy if you give me the right answer. Yes, Raman, you are perfectly right. Your concepts are very good. Good. So my answer, I'm writing this. So now you can see that there are four level of QC. One is for the negative HIV to ensure the negative reliability of your results. And the level two is a plus one, HIV-1 positive. Make sure that that the HIV-1 positive results are reliable. And the second positive is for the HIV-2 positive result, and the third positive is for the P24 antigen positive result. So the three positive and one negative QC helps to monitor the performance of all combo analytes which are detected in our HIV combo assay, right? So this is the brand about, excuse me, the SNIPE. We have a detecting fragment of HIV-1, HIV-2, and P24 antigen. We have a sandwich principle. We have a 50 test and 100 test kit size. We have a sensitivity of 100%. The specificity is 99.98%. So the HIV-1 P24 sensitivity is 0.7695 IU per ml. While the other competitors, their detection fragments are not available. In just some cases, they are available. They have a sandwich principle. They have a different pack size, 100 and 500. Their sample volume is 40, 100, and 300 microliters. The sensitivity is 100%, but the specificity is also lower in some cases. Like you can see that our specificity is better than all the other competitors. And not only the specificity is better, we have also the sensitivity for P24 antigen is better than the other competitor. So the our essays not gives the false negative results. So they gives more about the false probability that sensitivity is 100%. So the MAT can be checked by through the confirmatory testing. So if we compare the SNIPE controls and calibration and their interval, you can see that we have also edge in this area, that we have a negative control, +1 control, +2 control, and positive P24 control. We are free of cost and free of cost calibrator, which is at HIV-1 P24 high and HIV-1 P24 low. And the calibration interval is 14 days. If we compare with the other, they have to purchase the controls and the calibrators in some cases with the Roche. Roche is providing negative and positive calibrator free of cost, but the calibration stability is 7 days. The Siemens is providing free of cost calibrator, but the calibration stability is 21 days. But they have to purchase a control, that's the extra charge. The Abbott, they are using calibrator one, where they have extra charge. Their onboard stability is 14 days. So they have a negative, positive, and controls. They need to be purchased by the operators. This is the Backman who's have calibrators and control both are purchased, and their onboard calibration stability is 56 days. So in the terms of calibration controls, we are providing free of cost, and we have optimum calibration stability on board as compared to the two competitors, and the two competitors are have a higher than the our assays onboard calibration stability. So this is the assessment to the sensitivity on the basis of different groups and genotypes. They have detected 564 confirmed samples with the 100% sensitivity. And the assessment of the sensitivity also depends upon the 30 commercial seroconversion panels. And these seroconversion panels shows a more sensitive performance compared to the results, and gives you the results for other commercially available CE mark HIV assays. So we are excellent good in that area on the basis of different genotype assessment. The overall performance of SNIPE is detecting 530, 5,000, 3,000, and 5,360 negative samples with a specificity of 99.98%. So if we talk about the specificity, the seroconversion panel for the cross-reactivity, the panels are 109. We have a third-party assessment in a good agreement with the other competitors in the publication. There's some determination of seroprevalence, risk factor of transfusion, transmissible infection in the blood donor in Islamabad, Pakistan with Maglumi HIV. So this is the prevalence of transfusion transmitted infection among voluntary blood donor in tertiary healthcare facility in Pakistan. They have shown that the sandwich immunoturbidimetic acid is used in the detection of the presence of HBsAg antibodies, HCV core non-structural NS3 protein, and HIV-1p24, HIV-1 and 2 antibodies using chemiluminescence analyzers, fully automated Maglumi M800. So the seroprevalence detection is more important, and then they have done it in the countries where we have. So this is all about today's presentation. We have one thing which we need to be discussed is any question if you have, you can ask. Okay, just give me 2 minutes if you have time. For we are actually 22 minutes over. Okay, so I remember, Anton, you have a one question about. Okay. So everything is clear. Any question if you have, you can ask right now. Okay. If no question, I have to take a final picture. So we will close that training today, right? Okay, friends, if you have no question, we can close today's session. Okay, see you tomorrow. Have a nice day, and thank you very much for being with us and for your question. Thank you very much, and take care yourself, and have a nice day. Goodbye.